Hey everyone, and welcome to This Explainer. Today we're diving into something absolutely wild, the fascinating intersection of nature and computer science. Have you ever stopped to wonder how scientists actually managed to isolate the call of one single hidden animal in the middle of a screaming, noisy jungle? Well, we're going to look at the exact bioacoustic programming techniques used to synthesize and analyze those wild signals. Let's jump right in and see how just a few lines of code can uncover completely hidden biological sounds. So here's our game plan. We'll start with the bioacoustic detective story, move on to generating a simulated wild, grab our mathematical magnifying glass, execute the search, and wrap up by finding that hidden spike.
All right, part one, the bioacoustic detective story.
Think about the wild for a second. It's loud, right? Chaotic noise is literally acoustic camouflage. But code is our detective here. Check out this number, 44,100. In our source code, a variable called the sample rate is set to exactly this. That means to accurately capture audio, the code is slicing up just one single second of time into 44,100 individual data points. Honestly, it's a staggering amount of data to think about. And actually, it gets even bigger. The duration variable in the code is set to 2.0, so we're looking at a full 2-second window of audio. If you do the math, multiplying that sample rate by the duration, boom, we instantly have 88,200 data points to sift through for this simulated experiment. Moving right along to part two, generating a simulated wild. Before we can analyze our environment, we've got to build it, right? And the entire foundation of our digital lab relies on this one simple line: import NumPy as NP. Standard code would totally choke trying to process 88,000 data points that quickly. NumPy, though, it's specifically built to manipulate massive arrays seamlessly. It is essentially the heavy machinery of our bioacoustic lab. And this really shows what we're building with that heavy machinery. Our source material highlights a function called Generate Mock Bioacoustic Signal. Basically, it combines two totally different things. On one side, you've got a baseline environmental frequency. Think of your chaotic background noise, wind blowing, water rushing. On the other side, you have a rhythmic animal vocalization. This is structured, it's organized, it's purposeful. The function takes both and just smashes them together, perfectly hiding the animal rhythm right inside that environmental chaos.
Let's peek under the hood at how the code actually prepares to do this. We see t equals np.linspace. This line methodically maps out the time intervals for our simulated audio canvas. It starts at 0, runs to our 2.0 duration, and lays down enough evenly spaced ticks to hold every single piece of that massive audio data.
Which brings us to part three, the mathematical magnifying glass. Okay, so we've got our simulated wild environment, but now we need a specific tool to actually find our sounds. Enter the Analyze Acoustic Structure function. We take that massive audio signal we just generated, and we feed it into this new block of code alongside our trusty 44,100 sample rate. The entire job of this function is to dissect the complex audio stream. It's our digital scalpel. The really crucial part is how it dissects that signal. It uses something called a fast Fourier transform, or FFT for short. As the source defines it, this is a mathematical process used to extract dominant frequencies and spot organized, non-random spikes in audio data. Think of it like this. Instead of looking at a messy, squiggly line of audio volume over time, FFT translates that data to show us the actual pitch and frequency recipe that makes up the sound. It literally separates the random noise of the environment from the highly organized rhythm of our animal. It's the ultimate magnifying glass. Let's see it in action. First up, n equals len of signal just counts how long our data is. Then the magic happens, np.fft.rfft of signal. The code runs this Fourier transform straight onto our raw audio data. In literally a fraction of a second, it churns through tens of thousands of data points, mathematically pulling those chaotic frequencies apart to see exactly what's hiding underneath. But, a raw mathematical transform isn't quite enough. We have to translate it back into something human-readable. That's where np.fft.rfft freak steps in. It takes the output of our transform, factors in the spacing of our sample rate, and gives us recognizable dominant frequencies. It basically labels our findings, so we know exactly which pitches are present in our virtual wild. All right, part four, executing the search. We've built our environment and our magnifying glass. Now it's time to actually run the experiment. The final execution block breaks down into 3 really clear sequential actions. Step 1, Establish the rules by setting the sample rate to 44,100. Step 2, call the function to generate our mock signal. And step 3, pass that fresh signal right into our analysis function.
Here is the exact moment it comes together in the code. Time audio stream equals generate mock bioacoustic signal. With this one single line, boom, we have officially created our 2 seconds of simulated wilderness. The chaotic baseline environment is live, and that rhythmic animal vocalization is secretly injected right into the middle of it.
Finally, part 5, finding the hidden spike. Time for the payoff. Here is the final triumphant line of code. We take that audio stream, basically our haystack, and our sample rate, and we push them through Analyze Acoustic Structure. This is the precise moment the fast Fourier transform fires up, completely isolating the organized data from the chaotic baseline. When you step back and look at the inputs versus the desired outputs, the genius of this approach is just so clear. We feed it a messy camouflage signal and a simple sample rate. What the math hands us back are beautifully clean lists of frequencies and their corresponding magnitudes. We aren't staring at random noise anymore. We are looking at clear, non-random spikes. The animal's rhythmic vocalization has been entirely isolated and caught.
And that is the true power of bioacoustic analysis. We just watched a handful of lines of code perfectly generate a complex environment, hide a signal deep within it, and then mathematically rip that signal right back out from the noise. It leaves us with a pretty massive question. If this math can so easily isolate a simulated animal hidden in 2 seconds of code, what kind of unknown, hidden rhythms could it uncover if we pointed these tools at the real, untamed wild? The possibilities are honestly endless. Thank you so much for joining me on this explainer. Keep asking questions and keep exploring.